Hello, and welcome to our video where we describe the mechanisms behind the behavioral and cognitive experiences associated with Duchenne muscular dystrophy. Duchenne muscular dystrophy is a relatively rare yet extremely severe neuromuscular disorder that occurs in 1 in 5,000 newborns worldwide. Duchenne muscular dystrophy, or DMD, is a recessive X-linked disease caused by loss of function mutations in the largest known human gene, dystrophin, which spans about 2.5 million base pairs and has isoforms that are expressed in skeletal muscle and the brain. Normally, dystrophin is a long filamentous protein that serves as the link between muscle cells' cytoskeletal network and the extracellular matrix, providing critical support in stabilizing the muscle membrane, or sarcolemma, during muscle contraction. However, deletions and/or other mutations that result in the absence or near absence of dystrophin cause DMD and trigger instability in the plasma membrane and myofiber degeneration due to dystrophin's protein's inability to provide such support. The resulting symptoms are progressive muscle weakness and severe cell degradation or death that most notably leads to patients becoming non-ambulative in their early to late teen years, as well as a wide variety of other physical and psychological complications.
In fact, while the role of dystrophin in the brain is less well known than in muscle cells, research has confirmed that dystrophin protein is expressed in several structures that are involved in synaptic transmission and plasticity, both of which are critical for memory process. Therefore, mutations in dystrophin isoforms that are found in the brain have been shown to correlate with common cognitive impairments of DMD patients, learning disabilities, memory issues, attention deficits, behavioral difficulties, and even some neuropsychiatric disorders such as autism or OCD. Additionally, while cognitive impairments are common in patients with DMD due to lack of dystrophin in exceptionally important structures, the severity of the impairments experienced by patients with DMD is not universal and may range depending on the severity or types of mutations themselves.
To further understand the mechanisms behind the cognitive impairments caused by a lack of dystrophin, let's take a look at what current research suggests about the normal role of dystrophin in the brain, the genetic and molecular basis that DMD patients lack, and how this contributes to a psychological experience of this disease. Dystrophin has multiple isoforms, which are produced by the use of different promoters and alternative splicing. These isoforms vary in their size and function, and different truncations, such as DP71 and DP140, are named based on their molecular weight in kilodaltons. Here's an overview on how different dystrophin truncations are generated.
Dystrophin 427, or DP427, is the full-length dystrophin protein. It is 427 kilodaltons and is the isoform expressed in skeletal muscle. Mutations leading to the loss of this protein isoform cause the severe muscle phenotype seen in Duchenne muscular dystrophy. Other isoforms of dystrophin are generated through the use of an alternative promoter region. The dystrophin gene contains multiple promoters, each driving the expression of different isoforms. The use of these promoters is tissue-specific and developmentally regulated, which is why certain isoforms are seen in certain areas of the body and at different points during development.
The two truncated isoforms we will focus on today are DP71 and DP140. DP71's promoter is located in intron 62, and DP140's promoter is located in intron 44. Both of these isoforms play a significant role in the brain as they are widely expressed within neurons, localized to postsynaptic structures, dendrites, the axon hillock, and the cell body. This demonstrates their importance for neuronal function and synaptic connectivity in the brain and explains why there are cognitive comorbidities associated with some cases of DMD. The areas in which dystrophin is present in the brain are especially important for cognitive learning and memory, and mutations resulting in differing functional abilities of these dystrophin isoforms can lead to varying neurobehavioral output severities.
Let's start with dystrophin 140. DP140's promoter is found in intron 44. This isoform is primarily found in the cerebral cortex, the hippocampus, and the cerebellum. The cerebral cortex is the outermost layer of the brain and plays a critical role in higher-order cognitive functions, including reasoning, problem-solving, language processing, and sensory perception. In these areas, DP140 plays a role in synaptic function, neuronal signaling, and the formation of neural circuits. DP140 deficiency in the cortex is associated with the disruption of neural circuits that are responsible for executive functioning, leading to impairments in organizational skills, problem-solving abilities, and impulse control. Language processing and communication skills may also be affected by lack of DP140 expression if either Broca's area or Wernicke's area, two areas of the cortex responsible for speech production and comprehension, respectively, are affected by insufficient DP140. Another affected area by DP140 is the cerebellum, which is typically associated with motor coordination, balance, and motor learning. Deficiency here may also contribute to further motor balance issues due to inability to strengthen cerebellar motor connections. Lastly, DP140 is expressed in the hippocampus, a structure located deep within the temporal lobe that is crucial for memory formation, spatial navigation, and learning. DP140 is highly expressed in this area, particularly playing a key role in synaptic plasticity and long-term potentiation, or the strengthening of synapses essential for learning and memory formation. Individuals lacking DP140 may experience difficulties in learning new information, retaining learned material, and recalling past events, leading to deficits in both short-term and long-term memory.
Now let's shift our focus to DP71. While DP71 is primarily expressed in neurons like DP140, it is unique for its presence in certain types of glial cells, suggesting potential roles in supporting glial function, such as myelination, neuronal repair and support, and maintaining homeostasis in the central nervous system. Because DP71 additionally affects glial function, deficiency can lead to reduced synaptic transmission speed as it plays a role in myelination of neuronal cells. This isoform of dystrophin is widely expressed in the brain in the areas that are affected by DP140 as well, such as the cerebral cortex, hippocampus, and cerebellum, but also in areas like the basal ganglia, thalamus, hypothalamus, and amygdala. Its effects in the cortex, hippocampus, and cerebellum are largely similar to that of DP140, so let's focus on its expression in the other distinct areas.
DP71 is expressed in the basal ganglia structures, including the striatum, which consists of the caudate nucleus and putamen, the globus pallidus, and substantia nigra. The basal ganglia typically is associated with voluntary motor movement, reward processing, and procedural learning. Disruptions to the DP71 dystrophin isoform in the basal ganglia may cause difficulties leading to motor control issues such as dystonia or abnormal muscle tone, bradykinesia, the slowness of movement, and/or tremors. Patients may exhibit symptoms similar to that of Parkinson's disease, which also involves basal ganglia dysfunction.
DP71 is also expressed in the thalamus and hypothalamus, which are involved in sensory processing, regulation of arousal, and various other homeostatic functions. Its specific role in these regions is less well characterized, but may contribute to the cognitive and behavioral processes related to the sensory integration and emotional regulation. DP71 also affects the amygdala. The main role of this area is emotional processing. DP71 deficiency in this region could impact emotional regulation, potentially leading to increased anxiety, altered fear responses, and social behavior abnormalities. It has been speculated that these DP1 deficiencies in these areas may be linked to autism spectrum disorder in patients with DMD.
There is minimal research on therapeutic strategies to maintain cognitive abilities in patients with DMD, highlighting the need for increased awareness, funding, and research. Current speculation suggests that the most promising outcomes would include standard pharmacological interventions for DMD, such as corticosteroid treatment, combined with behavioral and cognitive therapies for treatment and management of the cognitive symptoms experienced by many patients. Further research should focus on the efficacy of cognitive enhancers such as standard of care ADHD medications and the potential for delivery of neurotrophic factors to support neuronal survival and function.
Overall, mutations in the dystrophin isoforms DP71 and DP140 have been linked to severe intellectual disabilities and behavioral difficulties. A null or truncated form of dystrophin in the brain can lead to a decrease in neuronal density and reduced synaptic transmission speed, impairing cognitive ability in those who express these mutations. Symptoms including hyperactivity, inattentiveness, learning and memory difficulties, and many other cognitive deficits continue to affect children with DMD regularly. Patients with DMD are at risk of developing these cognitive deficits due to their lack of functioning dystrophin protein. In the future, more research is needed to address these neurological symptoms and develop efficacious treatment strategies to manage this aspect of the disease.